I'm Ina, a biomathematician interested in networks. I think it's pretty cool that they can be applied in so many different fields. Currently, I use them to study marine microbial interactions. When you take one glass of water, it will contain millions of microbes. They are key players in major nutrient cycles. Did you know that on average, every second breath you take contains the oxygen produced by the microbes in the ocean? These microbes are important, but we don't know enough yet, and we would like to know who is interacting with whom. So-called high-throughput omics technologies advanced tremendously over the past years. We are able to determine who are these microbes, and even further, we can count them. Using these counts, we are able to infer microbial interaction hypotheses. When I think of microbes, I think of network similar to the social network. Each node is a microbe, and if two microbes interact with each other, they are connected by a niche. Microbes do not have a social media account. We can't just look up their connections. It needs mathematical methods to infer relationships between microbes. Why are networks so cool? For example, we can use them to infer important microbes. However, importance is relative and always depends on your definition. There is no ideal way how to detect keystone species of an ecosystem through a network. You could, for example, say that the important nodes are the hubs, that is, highly connected nodes. That would mean that in this example, the microbe of the crown is very important, and the microbe below it unimportant. This node has only two edges, but it functions as a bridge between two modules. So for network architecture, this node is very important, and it is up to the biological interpretation to find out how such network importance translates to the ecosystem. Speaking of modules, a module is a group of highly interconnected nodes, and then we would investigate why are these microbes more connected? Are they involved in the same nutrient cycle? Do they share the same environmental niche? So far, I told you that microbes are important, and networks are important to study microbial interactions. Now, imagine there is a network that contains all microbial interactions, a so-called meta-network. Each microbial ecosystem, at any given moment, under the influence of specific environmental selection, represents a subnetwork of the meta-network. I assume that a similar environment would likely lead to a similar subnetwork. If we gather enough knowledge how the environment influences the network architecture, we can later use that information to infer a network based on environmental data through a network similarity. That is the idea. My problem is, how do I obtain such subnetworks? I have several datasets. The first one is a time series of 10 years. One module marine microbial ecosystem in one location and one depth. The second dataset combines several samples across the ocean and depth layers. This dataset had 403 samples and is the biggest in the study. We all agree that sample size matters, right? But what about the size fraction used to filter the microbes? Does their size matter? Is there a difference between two size fractions? Maybe there's a difference if we look at bigger size fractions. And bigger ones. Dataset number 3 contained 5 size fractions, and the last dataset contained 2 types of data, microbial counts based on DNA and RNA data. These are my 4 datasets, each with a unique characteristic. 1 time series and 4 spatial datasets. When I started my project, the plan was to use microbial abundance data my institute already had, construct the network, and start analyzing them. However, the plan did not match reality. Instead of a pretty network, I obtained a hairball. A hairball is a dense network with way too many edges. So before network analysis, we had to filter the network. A simple threshold won't do the job. So we combined several filters, including environmentally driven edge detection. Don't hesitate to ask me later about it. And feel free to give me a comment, as this is my very first preprint of my very first author paper. And here you see the 4 networks I constructed. 1 network per dataset. Next, I want to determine sample-specific networks for each dataset. The problem is that co-occurrence is a needed, but not a sufficient condition to infer interactions. That means an interaction is not necessarily present only because 2 microbes are present. The solution I came up with so far is to connect the co-occurrence with the filtered association networks. I determined a subnetwork for each sample. Let me show you what I mean. The temporal data can be seen as a dynamic network changing in time. For each month, we now have 1 network. Here you see that the dynamic network seems to undergo a cycle. It's 10 years, and we observe a pattern and reoccurrence of the same edges. So again, how did I generate the sample-specific networks? In order to generate sample-specific networks, 2 conditions have to be fulfilled. The first one, an edge needs to be present in the original network. And the second condition, both microbes have an abundance above 0 in that sample. That's what I meant with the co-occurrence. I determined sample-specific networks for each of the 4 datasets. What I find fascinating is that we can now use them to study networks in time and space. There are several things we can do with them. For example, we can determine network properties. Here, I show edge density for the temporal network. Edge density is the number of edges divided by the number of all potential edges. On average, the edge density is higher in winter and lower in summer. Another thing we can now determine is edge prevalence, that is, the fraction of networks an edge is present in. For example, here you see the edge prevalence for the big spatial dataset. We found a low average prevalence. However, this is biased. Our dataset contained much more surface samples, and the surface samples have a high fraction of low recurrent edges. Having the sample-specific networks, we can also compare them to determine network similarity. And then we see how does the network similarity align with environmental factors. First, I measured network similarity based on the set of edges. For that, I computed the number of edges 2 networks have in common, divided by the number of edges that are in one or both networks. That is, the intersection divided by the union of sets. If 2 networks have exactly the same edges, the score is 1. And if 2 networks share barely any edge, the score is close to 0. For the temporal data, the average similarity was low, but it increased when we compared networks intra-seasonal. Our results here point to a seasonally dependent reoccurrence in network architecture. It is ranging from modest to moderate, and we found highest similarity for winter and lowest for spring. We can use the network similarity scores to construct a network of networks. Each node represents one of the subnetworks you saw in the video. And if 2 nodes are connected, it is because they are similar according to their edge set. Since each node is one sample-specific network, we can map environmental data. Here, I use temperature because it has been shown to be a major factor driving microbial abundances. As you see, and as expected, it aligns pretty well. Low temperatures are with the winter months all connected to each other, and when we go through the network of networks passing by the spring months, we reach the warmer months, which are the summer months in our time series. I use the same approach for the spatial data. Also here, the network of network aligns well with temperature. It also aligns well with depth. This is expected since temperature and depth co-vary in the ocean. The effects of depth and temperature override the effects of latitude and longitude. That is, if you take two networks from two stations that are different but the same depth, these two networks will be similar. And they will be more similar than when you would take two networks from two depths of the same station. We also constructed network of networks for the 5 size fraction dataset and the DNA RNA dataset. There were no surprises. All 3 spatial datasets indicate that network similarity aligns well with environmental similarity, which means if we gather enough subnetworks and the environmental data, we may be able to use them to predict a network based on environmental data and microbial composition. This is based on the meta-network idea I presented to you in the beginning. I showed you that edge prevalence was higher for deep ocean layer than surface layer. By the way, it was also higher for winter than summer months, and they are similar for different size fractions as well as DNA and RNA data. We saw that network similarity aligned well with environmental similarity, which is a first indicator that microbial interactions are affected by environmental selection, but also that we can use environmental parameters to predict networks. Coming back to the meta-network, wouldn't it be cool to be able to do predictions of subnetworks for a specific environment and microbial constitution? Thinking of climate change and the global warming, we could predict the influence of rising temperatures on the microbial interactome. The problem? Extrapolation bears uncertainty. However, we may lower uncertainty by combining different types of data. Just think about genomics, transcriptomics, proteomics, metabolomics, single-cell data, all combined in a multi-layer network. And given the advance of technologies and some supercomputers, I think we are already going in that direction. The ideas I presented to you today were impacted by many people, their support, and fruitful conversations. I want to acknowledge especially Ramiro Lugares, my supervisor, and his former postdoc Anders. I did 2 research stays, one in Belgium, where I was hosted by Caroline and Gypsy, and one in France with Damien and Sam. There I met Marco and Erwan, who helped me with the network video. Thank you for your attention, and let me know how a network of networks could benefit your study.